Hi folks, I hope this finds you well. My name is Meredith Price, the Imaris Business and Product Manager. Today I will be giving one of the last Imaris Homeschool webinars, and this one is going to be about our new classification tools and the multitude of applications that are possible now with it. I am going to go in and make sure that you have a really good understanding of these classification tools. And so first I'm going to start off by giving you an introduction to some of the classification applications. So different types of research and different types of questions that can be answered using our classification tools. And then I'm going to make sure that you know the differences between our different types of classification. I'm going to do this partially within PowerPoint and then partially within Imaris as I'm giving live demos of some of the applications that I go over. I just want to make sure that everyone knows how to find the tools and when it's best to use one tool over another. And then finally, I'll go over how you can use these tools when performing batch processing to make that throughput for your image analysis that much faster. And then finally, we'll have a live Q&A like we have with the rest of the homeschool webinars. As I get started with going over these applications, these again are really just a taste of what's possible. And we know as biologists that performing classification and separating what we see within our images into various groups is incredibly important for us to be able to tease apart the differences within our images and within our experiments. And so, again, these are just a few examples. The first one over here on the left-hand side is an example from the immunology space where we have some vessels that you see in this yellowish color, and then dendritic cells are labeled in the pink and the cyan. And in this case, we're wanting to look at a gauge of cellular affinity. So how many of these dendritic cells are within a certain distance of the vessels? And so because we've been able, in this image, we've been able to create these objects to represent them, now we understand where they are within the space of the image, and we can automatically generate these distance measurements. And so so we're looking at proximity or distance measurements, and we're able to say that the dendritic cells that are within 5 microns, we have a certain number within this image, whereas we have a certain number of pink cells that are further away. We have almost a completely different type of question in this example in the middle that might be one that you would see within cell or cancer biology, where we have these protein structures that you see in the raw image or the raw signal within blue, but we've also detected some objects within these hexagonal structures or just within the center of the hexagonal structures. And we've been able to specifically pull out those and then classify, we've been able to specifically pull out those that are in the center and then classify them based on their shape using a machine learning classification algorithm. And so this is an example where we're able to grab these protein aggregates based on their location, their shape and their size, and we're able to detect them and then classify them so that we can really narrow down our focus on the question that we're trying to ask. So that's another example. And then over here on the right-hand side, we have a sort of a classic image from developmental biology where we have an early embryo that's beginning to set up its body plan. And so we see that patterns are starting to form in the shape of these stripes that you see within pink. And we have nuclei within the cyan color. And you can imagine that you have experiments where you're wanting to test the fidelity of this pattern formation, of this striped expression, and that you need to count cells. And that's something pretty basic that we see not just in developmental biology, but elsewhere where you're looking at expression. And this is a place where being able to classify based on this intensity is incredibly useful. And and the reason that I put this asterisk beside intensity is that if I were, and I'll show you this in the live demo in a little bit, if I were to zoom in on this image, unfortunately, this protein expression is not within the nucleus, and that's what we're using to identify all of the cells as that nuclear label. And so it makes it hard to use that and that protein expression to classify the cells. But because of some nice new tools that we have within Imaris for the machine learning classification, we are able to do the classification that you see at the bottom there. Some other examples are cases where maybe you're a researcher that is looking at disease states or something like that, and you're looking at whole organs or larger tissues. And so we have that over here on the left-hand side, where we have this complex tissue where getting, again, something as simple as cell counts per type of cell seems straightforward, but when you have thousands or tens of thousands of cells, it can be quite burdensome. And so something like automated classification can really improve your throughput and make your life much easier. And so in this case, I'm just going to show you that the result of this, where I've classified based on 3 different generic cell types here. You could obviously do more, and again, we're using intensity as well as density of cells to be able to do this classification in an automated way. Another example from the world of cell and developmental biology, and I have to admit, I give myself a hard time that I do love Drosophila, so you're seeing another Drosophila embryo. This is a case where we have specific cells that have differentiated, but I, in this case, wanted to grab the cells that are labeled with this orange label, but just the cells that are distal and are along the motor neurons, and not the ones that are along the midline. And using a tool that we have within Imaris called reference frame, I am able to define that midline and then use that measurement, this location or distance measurement, along with intensities, to go in and be able to classify those orange cells and find them separate from the ones that are in the midline or all of the other cells that are identified within this embryo. And so again, classification can help you very specifically identify subpopulations within your tissues, or subpopulations within your protein aggregates, that sort of thing. And then finally, again, a number of different disease states might be looking at something like wound healing, and this is a common image analysis example where being able to go in and find just the cells that are the ones that are crawling and not the ones that are stuck to the bottom of the plate, or things that aren't actually cells but are bright spots, that sort of thing. That is a common problem, that you want to ensure that your data is as high of quality as possible, that all of that tracking information that you get from your time-lapse analysis is good and can be used. And so one of the first things you really want to do is make sure that the cells that you've identified, that you can classify those into good and bad cells. And so that's one thing that we've done here. And again, that's done based on their location. The ones in the center aren't moving, or they're stuck to the plate, whereas also the ones that sort of are on the edge but are not as bright, we aren't being detected either, or aren't being classified as good, excuse me. And so these are just a few different examples of applications where you can use object classification. But again, this is not a comprehensive list by any means. And so I would recommend that if you are interested in using these tools and you're already an Imaris user, please reach out to the team. We'll definitely be able to help you and talk you through it, and specifically for your images. If you're new to Imaris and you want to check it out, again, this is a place where we can help you, and we'd be happy to both offer you a demo version of Imaris to use as well as to work with your data sets. So from here, I'm going to go in and tell you a little bit more about the different types of classification tools we have in Imaris just within this PowerPoint, and then I'll move in to the live demo within Imaris. So the first type of object classification in Imaris is a manual method, and this we really had already, and we called it labels, and it was in the bottom right-hand corner of the Surpass view within Imaris. So that's still available, but now you get to it by first going through the object detection creation wizards within spots, surfaces, cells, and filaments, and then accessing the labels menu within a new edit labels tab. And so that's this icon that I have here on the screen. And with manual editing, it's nice to be able to have the ability to edit any of the classifications that you've made and the other more automated methods, but also to be able to completely generate classes de novo on your own. And that can be useful when you just have a small amount of objects to classify, or if you're using it as a tool for labeling other things, as maybe you're editing tracks or that sort of thing. And so you will find it useful, and I'll go into that when we go into the demo within Imaris as well. And then we have two different types of statistics-based ways of performing the object classification and doing this in an automated way. So these are going to use histograms of the statistics that we're already offering within Imaris. And so one is using just a one-dimensional histogram, so just one statistic. We have another method that's a 2D histogram. And so both of these are found in the same place, and they're found within the creation wizards for spots and surfaces. And you are able to go in and set up almost as if you're setting a rule where you're picking a statistic and then picking values to create the groups within a set. These can be layered, so you can take a group that's been defined and or a class that's been defined, and then separate it even more and divide it up into even more classes. That's one thing that you can do. You can also use these classification methods or the statistics-based method within batch processing. And so I'll go over that at the end. And then finally, we have a machine learning-based method for performing the object classification. It uses a random forest classifier, and again, it's found in that same place within the creation wizard. It's also available for spots and surfaces. It can be layered and used with those statistics-based histogram methods that I mentioned before, as well as with batch processing. One thing that's useful to know about the machine learning is that it gives you access to extra statistics. And so one way that we're able to accommodate, and I'm going to go back really quickly, accommodate labels where the intensity is not within the object, but maybe around it and outside of it, is because we have included these extra intensity measurements, as well as a few, as well as some other measurements, and given the random forest classifier access to those. And so I'll show you where you can see when it's using those extra measurements, but it's incredibly useful and can give you this extra reach and an extra ability to analyze data sets that you weren't able to analyze before. Okay, so now I am going to go into Imaris. I'm going to take some of those examples that you saw in the PowerPoint slides previously, and I'm going to show you exactly how I went about analyzing them. So just hold one moment while I switch over. So I've opened up Imaris, and you can see that I've got Arena showing you some data sets, and hopefully you recognize that early embryo that's in the process of pattern formation. And so I'm going to open up this example right here, and we can see, as I had mentioned before, that we have some nuclei labeled in cyan, and then we have this protein expression in stripes that we're seeing in this pink channel. And what I want to do here first is to go through and show you how you would go about performing the manual classification. So I'm going to go in and actually by hand label some of the cells that are within the stripes and show you how to do that manually. And then I'm going to go back and show you how I would do that using the machine learning tool. With another example, I'll get into the other types of classification where we're using the statistics-based histogram methods. Okay, so let's get started. The first thing that I'm going to do is to add a spots object. And so this starts the creation wizard for spots. I am going to use, again, this nuclear label to find all of the different cells within this embryo. Okay, so now I'm going to just get started going through the creation wizard. Using 4 microns is perfectly fine for my spot size here. So now Imaris is going in, and for that cyan channel, it has found all of the different nuclei, and the automatic quality threshold works pretty well here. So we can just keep going. This is where this classification step is, but the first thing I'm going to do is show you how to do this manually, and you don't do this during the creation wizard. So I'm just going to hit the trash can button to get rid of the classification. You can see that the color of the spots now goes back to that gray color. And now I'm going to hit the green button down here at the bottom of the creation wizard to finish up the wizard. Okay, so now it's completed. We have our series of tabs, and the one that I want is that sort of yard sale tag, let's say, with the green and the red tags. And what that does is it takes me back to something that looks a lot like what it looked like with what that classification step looked like as we were stepping through the creation wizard. And I'm going to hit the plus sign to add a new set. It's going to ask me what I want to use as my input. And yes, I want to use my points or my spots. And then I can set up 2 different classes in this case. And so I can say that I want to have the cyan class for all of the nuclei that are outside of the stripes, and then I'm going to actually go in and change the color by clicking on that green square, and I'm going to change the color to pink and just say these are the nuclei within the stripes. Okay. So now how do I go in and make the assignments? Well, one thing that's really helpful for you to know, and I'm just going to get rid of my display adjustment window for now, is that you can switch over, and you can do this with your mouse. I'm over here in the upper right hand corner. You can switch over from a normal selection mode where you're just clicking on one object at a time to one where you're selecting multiple objects at a time. You can do that with your mouse again, or you can use the escape button to toggle between those. And what this means now is that I'm in circle select mode, and I can select a bunch of objects at once, which is super useful if you're going to do something like manually classifying or labeling objects. So I can hold down Control and grab a bunch of them, so I could go through and pretty quickly, with a decent amount of accuracy, go in and select all of these guys at the anterior end of this embryo. And so then I'm just going to hit assign. We can see that that was 580 spots that I selected. And now I'm going to go through, and to make this a little bit easier on myself, I'm going to zoom in using the scroll wheel. Change the size of my circle select tool. Again, I'm using the scroll wheel but holding down Control. And now I can go in and select some of these. may need to be a bit smaller. But hopefully you see that although it's not so hard to do it manually, actually getting it absolutely perfect can be a little bit tricky. So having some help and having a way of doing this, automatically is super useful. Okay, so I've gone in and done that. I am actually going to turn off the volume so that you can get an idea of what that looks like now. So we have those cyan colored spots and the pink spots, and I missed one guy right there. Okay, so now I do have those counts. If I go over into the statistics tab, and this will be true if we use the other methods of classification, when I go in and look at, let's see, values like distance to nearest neighbor, that's a new measurement that we have within Imaris, I can see that for this set, so it's set 1, where the cells have been classified as either class B, I'm going to sort them, or as falling within stripes, that is added to the statistics table. So when you look at it within Imaris, or if you export it out and work with that data within Excel, all of that will be there for you. Again, if you wanted to change one of the classes, you can always select some of these cells. You see they're selected over here in the Stats tab. You can go back over here and you can assign them differently if you need to do that. So you can go in and edit something that's been done before. Okay. So that's how you would use the manual classification. Now what I want to do is actually turn this off and turn back on my volume and show you how much easier it could be for you. So first thing, again, I'm going to go in, I'm going to make sure that Classify Spots is checked on, because I want to make sure that that is available as I'm stepping through the creation wizard. Again, I'm going through this object detection step. It works quite well with the default size and with the default threshold. And then I get to the classification step. And in this case, I don't want to use the 1D or the 2D histogram methods. I want to use the machine learning method. And in this case, it's pretty similar in that I need to select cells and assign them to a class. That will then make up my training set that is fed into the random forest classifier. Now, I was thinking about this as I was hanging out with my daughter, and it's one of those situations where, you know, sometimes we say, do as I say and not as I do. But when you're training the random forest classifier, you need to make sure that that whatever you're giving it is really what you mean and really what you want it to do. So just like a 4-year-old who is listening to everything. So just imagine that the classifier is listening to everything and it's paying attention to everything that you're training. So make sure that that training information is as good as you can give it. You don't have to give it a ton. You'll see I won't give it too much, too many objects for the classification, but that I want to make sure it's high quality information that I'm giving it. So again, I'm going to use that circle select tool. I am going to assign the spots that are in the anterior region, that are in between the stripes, and then that are at the posterior region. I'm going to give them this blue color. So I'm going to make it a little bit bigger. I'm going to grab those two. Those are easy. I'm going to go ahead and assign those or classify those. And then I'm going to zoom in, make my circle a little bit smaller. And I don't want the model to learn anything about, the location, let's say, so much. And so I'm trying to scatter some of these spots around. And you want to make sure that, in this case, we have thousands of spots in these cells. And so you want to make sure you give it a good amount of spots to think about. You can tell I'm thinking as I'm selecting these. Okay. So I assign all of those, and now I'm going to go in and grab some of the spots for the stripes. Because I'm not doing this manually, I don't have to be as perfect as I was trying to be before when I was doing it manually, but I do want to scatter things about. And so I learned how Imaris best performed on this data set just by playing with it a little bit. It wasn't too difficult by any means, but sometimes you do have to play around just a touch. Okay, so we're going to hang out as Imaris is feeding in all the different statistics that Imaris already in previous versions had calculated, as well as some new intensity statistics, as I had mentioned, are being calculated. So now that's done. And with the colors I chose, it might be a little bit difficult to see what the results look like. So I'm going to turn off the volume. And now you can see just the spots and their colors. And it's pretty good. There might be a couple places that I don't completely agree, like right in there. I think that those really should be pink. And so I just selected them, assigned them again, and we'll do train and predict, and I got a little bit different of a result. After you, as you go in and refine or iterate on your model, on your classifier model, you will notice that it goes much faster than it did before. Okay, so now I have this result. I am happy with it. I can finish my creation wizard. I can go again to this edit labels tab. I can see my counts here of the number of nuclei per class. I can also go and see that in the overall tab within the statistics tab. And what I can also do is go over and see these within Vantage. So Vantage is our plotting view. And the first thing that you'll see is that all of these spots are being shown in a 1D dot plot, and it is using the average distance to the 3 nearest neighbors. That's one of our statistics, and so we've grabbed that one first. But what I want to do is actually hit this link or like chain, and so it breaks that up and shows me the two different classes that I had before, and now I can compare those and scroll through in this drop-down menu to different statistics and see how those compare across the different classes. And so you can do this within just one image, or if you did batch processing or not, you can look at this across multiple images as well. Okay. So there was that first example of performing machine learning classification in this developmental biology, pattern formation, cell counting type example. So now I'm going to go over and show you how to do this in another data set. The next data set we're going to look at is the one from Immunology. And so in this case, we have vessels that are in this orangey yellow color, and we want to know how far away are the dendritic cells that you see in this lime green, how far away they are from the vessels, so that we can classify them and then see if there are any differences in count or other characteristics between those two groups, ones that are closer versus the ones that are further away. So the first thing that we need to do is we need to set up our reference. We need to create a surface for that orangey vessel so that we can measure those distances. So to do that, we are going to use a surface object, a surface reconstruction. Now we aren't classifying it, so we can turn off that classification step by unchecking this checkbox here, but we will need to make sure that we are calculating the shortest distance, and so that is checked on. I'm going to go through the steps of the wizard and make sure that I change the channel. I'm bad about that. But I am able to use some of these default values to go ahead and make a surface pretty quickly here. lower that threshold just a little bit. Okay, so that looks pretty good. I'm going to go ahead and finish up my creation wizard for the surface, and now move on to those dendritic cells, which we are really interested in. In this case, I am going to use a surface reconstruction for them because they do have a bit of an odd shape, and so I want to get those contours, and I can do that with surfaces and not with spots. Now, as opposed to last time with vessels, I do want to classify the surfaces, so that is checked on, and then again, I need to make sure that Imaris is calculating those distance measurements for me, so that needs to be checked on as well. I'm going to step through the wizard. Again, I can use these default values. They work pretty well in this image. And Imaris finds all of these dendritic cells pretty well for me. Just make sure I grab a couple more. Okay. It does a quick filter. And now I am in this classification step. So how does it work? And when we are using the histograms? In this case, I'm only caring about or assaying for the classes. I'm only worried about the distance from the surface that represents the vessels. And so it's just one measurement, so I'm just going to use a 1D histogram. What I need to do is go in to this drop-down menu and find the measurement that I want. So you can see I have all of the measurements within Imaris surfaces checked on and available in this drop-down menu. Excuse me. So you have a lot of different things to choose from. What I'm looking for is shortest distance to surfaces, and it's to that surfaces 1. So that's what this one up here. So now you can see that the dendritic cells, the surfaces that represent them, and I can make this a little bit more clear for us by turning off the original data. You can see that they're color-coded based on the ones that are further away are pink, and the ones that are closer to the vessels are blue. But I can adjust this, and you can see as I move this threshold that is defining the two different classes, you're seeing the value pop up here along the line. Now, I want to have it so that it is at about 5 microns. And so what I can do is with my scroll wheel, I can actually zoom into this histogram. You can zoom into any histogram within Imaris, and then I can get a bit more precision here. So right around 5 microns, I've set it up my classes, and you can see that that they're color-coded within the image this way as well. Now, if I wanted another class, all I would have to do is add that here. You can see this green class. Again, you can click on here to add a color. And I need to zoom out to see where my green class is. Yep. And so now I can adjust that in here too, if I wanted to. In this case, I'm going to get rid of it. I just want these 2 classes, and now I can finish up my creation wizard and move on over to the vantage view if I would like to be able to again check out a dot plot looking at the the 2 different classes and the surfaces. So the ones that are closer and the ones that are further away, and looking at their surface area measurements to see if you see a difference, a significant difference. Now, we don't have very many objects in this case, so but it does start to give you an indication of how they compare to one another. Okay. Again, you can find you can find the counts, some rough information within this Edit Labels tab. You can go in and change the, you can change the colors if you want to, and change their names if you like to as well within this tab. And then you can also change the visuals or the coloring, or the appearance of the classes within the color tab. So I can actually turn the labels off if I want to, or only show one of the classes as well. Okay, So that's a pretty straightforward example of using just something, using a distance measurement and just using one measurement using with the histogram or the statistics-based method to do your classification. So now I'm going to go in and actually show you another method where we're going to use distance and intensity and use the 2D histogram to perform the classification. Okay, and so now the next data set that I want to show you is one where we're going to be looking at cell differentiation, where we want to pick out specific cells based on not just their expression of a certain protein, but also because of their location within the embryo. Okay, so let's open that data set up. In this case, we've got a few different channels. I've just got the one showing the expression we care about in orange, as well as the nuclei labeled in this pink color. And then also I want to point out that I've got something here called the reference frame. And what it's doing, and so it's sort of like a tripod here, is that I've already placed it within the data set, and it is lined up with the midline of the embryo, so that it's defining a plane for me where I can say that the y-axis is going in this direction, and it'll help me to be able to exclude the cells that are labeled in orange that are at the midline within this big chunky area right in here, because I just want to grab the cells that are out here. Okay. So now, again, I am going to, similar to what I did in the first data set, I'm going to identify the cells based on that pink label and go through the spots creation wizard. So I'm going to add a spots object. This time I am going to classify these spots. So I'm going to go through the wizard. These 2.3. And the resolution in Z on these isn't that great, or in this image isn't that great, and so that's why I'm looking for spots that are elongated within the Z-axis. So this threshold is pretty good for this data set here. And now I've moved on to the classification step. So in this case, I don't want to use just a 1D histogram. I need to use two different statistics that Imaris calculates to find these cells. You might be able to use machine learning to do this, but I found that using the 2D histogram was straightforward and easy enough to do. So I think it's a good example of a place where sticking to just a rules-based method with specific measurements is a perfectly good way of going about the classification. So under filter type here, in this drop-down menu, I'm selecting filter 2D, and now you're seeing a heat map of the number of objects, and so that's why you see the change in color. And so now the top measurement within this drop-down menu is for the x-axis, and the bottom measurement is for the y-axis. I want to go ahead and use that y-axis from my reference frame on the y-axis of this plot. It'll help you look at things and make sense of stuff if I do it that way. And then I want to use an intensity measurement, so the average intensity for channel 2, which is that channel that's in the orange color. And so now what you can see, again, remember that we have that the colors here within the histogram are giving us like a heat map for the number of the spots that are found in any given area. And so what we're seeing is that along the y-axis, we have a good number of spots clustered here. They are not very bright because remember on the x-axis, we're looking at the intensity measurement. And so we don't want the ones that aren't very bright. We also don't want the ones in the midline, and so we are seeing a cluster of these in the midline. And so instead, I'm going to grow and make this pink class nice and big. And if I turn off the volume, you see I'm grabbing the brighter cells that are or brighter nuclei that have this orange intensity, this orange label. I'm picking those up, but only the ones that are further away from the midline. And you can see how I sweep in if I move this pink box within the 2D histogram. So everything else that's not within the pink box will be within the blue class. Now I need another class because I need to grab the ones that are over here on the other side of the reference frame, on the other side of the midline. And so I'm going to add a new class. These are going to be green. You see that pop up. I don't want the dim ones. I just want the bright ones. And now I can interact with that box to grab some more. Pull this back a little bit, and I've grabbed these few. There's it doesn't grab this as much of this side of the embryo as it does of this side, so we have fewer over here. Okay, so that's pretty good. I'm happy with that result, and now I'm going to go forward, and I just have to finish up my creation wizard. I can go over here to the Edit Labels tab to get my quick view of what the counts are. I can see that for the blue nuclei or cells, I've got over 6,000 of them. For the ones that are that were the ones that I put into the pink class. So again, these are the ones that are labeled with orange. And if I turn off that, you can see a bunch of them picked up. I've got almost 500 of those and something like 63 of the ones down here. Now, if I wanted to, I could go in and fix up some of the errors that I can see a couple down here, and I could fix up those manually if I wanted to. I can also go in and compare the groups, the blue group to the other two groups within Vantage if I wanted to at this point. So I hope you saw from this example, though, that you can go in and use that 2D histogram to look at two different measurements at the same time and see that heat map and be able to, in an interactive way, create those classes. Okay, so then I'm going to have one last data set where I go in and show you an example of how you can use two different sets, so two different classification steps almost, and layer them together to get exactly what you want. Okay, so in this example, what we're going to look at are some junctional proteins. And in this case, if I zoom in, what I'm seeing is that you can see these proteins that are in these hexagonal rings, but I'm also seeing these protein aggregates in between or within the center of these hexagonal structures. And I want to characterize those and see if I can learn more about them. And so to do this, because they're this oddly shaped thing, I'm going to characterize them by using the surface object. So the first thing, again, I'm going to add a surface object. I need to make sure I'm using classify surfaces, and I'm going to just go ahead and leave shortest distance calculation checked on because I'm lazy. It actually doesn't make a difference here because I'm not comparing the distance with another object. Okay, so now I am going to adjust these values a little bit. I've obviously worked with this data set ahead of time, so I know that the size of the objects I care about is about 0.6 in diameter. So this data set isn't huge, but there's lots of little objects within it. So it took a second for Imaris to show me this preview, and then I'm using the interactive preview to help me pick a threshold that will work well for the object detection. So what can be difficult about finding these little protein aggregates that are in the middle of this hexagonal array is that this hexagonal array is actually curved, and so as you zoom out, it actually looks quite messy around the edges, but it isn't so messy as long as I rotate and tilt the image. And so this is a place where first going in and doing a bit of filtering, then doing a bit of classification, and then finally some machine learning classification can help me get to the point where I get my answer relatively easily. So now that I've defined where my threshold is, I'm going to move on to the next step. Nice, pretty surfaces. Okay, so now what I want to do is go in and say, you know what, I don't need the big stuff. I'm only looking for these little guys. So I'm going to do some filtering to get rid of some of the bigger things. So let's not make Imaris work any harder than it needs to. So I don't want to get rid of too much because I want to make sure that I keep any of the protein aggregates that I find in the center of these arrays. And again, remember, if you need to have more precision, you can always zoom into your histograms. So I'm doing this down here. Okay, maybe gone a little bit too far. Another thing that I can do is not just to filter based on the size or the number of voxels that fall within these surfaces, but I can also do it based on where they are within Z. So if I turn this image on its side, you can see there's actually some depth. No, I don't need these guys that are back here. So I'm going to add in another filter. This is not classification, this is filtering. And so that means that if it's not within the yellow part of the histogram, it's getting thrown out. But that rule that we always say about garbage in and garbage out kind of applies here too, in that let's at least not make Imaris work any harder than it needs to. Okay, so I'm going to keep everything at the bottom and get rid of the stuff at the top. Okay, and that went bye-bye. Okay, so that's pretty good. Okay, so now I'm going to move forward to the classification step. The first thing that it wants to do, it's a little tricky until I get it head on for us. The first thing that it's going to do on this classification step is to show you a 1D histogram. In this case, I don't want to do that. I want to do machine learning. So I want to go in and get Imaris to first go in and say, these protein aggregates that are on the inside, these are the ones that I want. And so I need to tell it what I want, and I also need to tell it what I don't want. And so I need to make two classes. First, these are the guys that I want. I'm going to go to another part of the image just to make sure it doesn't learn any position rules or sort of traits inadvertently. Now, I could try and teach it both types of classes that I want right now, but sometimes it's just more straightforward to take it a step at a time. And then, okay, so all of those are things that are in the center, and then I am going to say, okay. And yes, I talk to Imaris sometimes. Don't. And then I'm going to go in and start labeling and assigning protein aggregates that are right along these hexagonal arrays. And this is the stuff I don't care about, and I don't want to get into my final counts and into my final classes that I really care about, that I want to compare against each other. Okay. And so once I've done a good amount of each of those, now I can hit train and predict and have the machine learning classifier go through and try and pick up which traits are the defining measurements that help me pick out what I'm going for. And so hopefully what you can see is that pretty quickly, Imaris was able to go in and pick out as the blue aggregates the ones that are within the center of this hexagonal array. So it's grabbed those for me, which is great. It's awesome. That's what I needed, right? Now I did this pretty quickly. I'm sure there's a few errors in here. If I spent a little bit more time, more than the like 1 minute while I'm in the middle of a webinar, I could get an even better result. Okay, so now I've got the blue ones, and I want to take the blue ones and I want to split those into 2 groups. And so hopefully you can see there's some that look like doublets or like budding yeast, like a mother and a daughter, something like that. Whereas there's others that are almost perfect spheres that are separate from one another. And so I want to put those into 2 different groups and compare their size and compare the number of them and maybe even their location, their position within the image. And so what I'm going to do is add another set. And this set is going to be made up of, I need to go back. So I can. So I need the pink class, and so it's class, I need the blue class, and that's class A, see? But it's all there for you, even if you get tripped up like me. Okay, so I'm going to go in, and I need to have the input be class A. And so now what you're seeing is that everything that was blue has now changed colors into either green or to red. And the other one, the rest are that pink color. So I could go in and use a 1D histogram using a measurement like volume to do the classification if I wanted to. And so the bigger ones are red, the smaller ones are green, and I could be done then, and that would give me a difference in count. But I'm for sure doing it based on volume. If I want to study volume, I might want to do my classification in a different way. So of course you could go in and use a different measurement for this 1D histogram, or you could go in and do machine learning, where I'm saying, okay, that's for sure a doublet. That's a doublet. Now, these most likely are also bigger, so that might be what the machine learning algorithm picks up on. But again, I am also feeding into it different characteristics like shape and not just volume. Okay, so now I can go in and grab some of these other ones that are smaller. You can see me going ahead and doing that. And I am going to label them and as the red class. So within set 2, these are class B. You can change the color and change the names. Don't forget that. So if you're having trouble keeping track of things like I did, you can go in and name them appropriately too. Okay, did not mean to have 20 and 20 for each. So now I'm going to go in and train and predict again and zoom back out and see if it's done a pretty good job. And I think it has. There's a lot going on, so I'm going to zoom in. But hopefully what you can see is that most of these have been classified correctly. And when they're big, they're green. There are some errors. Again, I didn't spend as much time as I probably should because I'm trying to show you an example pretty quickly, but it's done a really good job of classifying these given the amount of time I've spent. Okay, so then again, once you're done, you can go ahead and finish up. One thing that I definitely want to show you now is the fact that you can change the visibility of the objects once they've been classified. And so to do that, you go to the color tab. And here, I really just don't care about this class B. I don't want to see them. So I'm going to uncheck it. Now, if I just do this, they turn into this, back into this gray color. But if I check on only show labeled objects, they disappear completely. And so that's what I really want. And so now I'm going to actually turn off that blue color because it was making things an odd color. But what you're seeing now is that the only things that are left are the green ones and the red ones. And I've got these nice groups with mostly objects found within the center of these hexagonal arrays. And so that's a method where you can go in and use different types of classification and layer them together to be able to get the result that you need. And okay, so the easiest way to set up your batch pipelines is to go ahead and use new batch and go through the creation wizards there. You do have the classification steps available within there, and then you can apply it to all the different images that you have. From there, you can go in and make plots where you can compare classes within images and across images or across groups of images too. So you can go and have fun with that and save yourself tons of time. Okay, and then with that, I think we will start to take some questions. So give us 1 second to pull up those questions that have been coming in. I know Anna has been answering them.
Okay, so hopefully you all are, you're seeing my PowerPoint, and so I just wanted to make sure that I was giving thanks to the few labs that offered us some of their images to use in the webinar, and as we're showing off the new features. So thank you so much to the Pfeiffer lab and the other labs within the Netherlands for helping us with that. And if anyone has missed any of the webinars so far, I do want to remind you all that you can go in to the Learning Center on our website. So it's imaris.oxtents.com, and then go to the Learning tab. And if you type in homeschool, you'll get all of the webinars that we've had so far. So there's going to, I think this is number 18. We're going to have one more. And so that's going to be given by one of our developers, Sasha Geyer, next week about how to write your own Imaris extension. And we've talked a little bit about those extensions in previous webinars, not so much in this one today, but in previous webinars. So definitely go and check that out. Okay, so we've had some questions come in, and looks like we've promised a few people. Yes, and we've promised a few people that we will answer their questions. So I'm going to just switch over to Imaris now. That's what you should be seeing, and I want to give you an idea of why some things are set up the way that they are, and then also how to see some extra information that I promised and then didn't get to during the webinar. So the first question that I want to address is, does Imaris actually use all of the calculated parameters? I made a really big deal about all of Imaris having access to all of these measurements to be able to use during the machine learning, but is Imaris actually using those? And actually, it's not. And what you can do, hopefully you see now, I'm going through, just redoing the work I had done on this Drosophila embryo and going through the creation wizard. And what you can see is that if I switch over away from the train tab, but over to the features tab, I can actually see which of the statistics, and some of these are ones that are new statistics, like this one here, and this one's new and this one's new, versus existing intensity statistics that we have. And it's showing us which of those measurements are being used and the weight of them. So some of them are weighted quite heavily, whereas other ones aren't. And if you, for some reason, and I have to say, most of the time you have to be really digging in to go into this. If you, for some reason, don't want one of these statistics, or as we're calling it here, features to be used, there are checkboxes here that you can change. You can look up the names of some of these statistics if you're interested to find out more. Most of them are looking at intensity values within the image away from the actual object. And so it's using that information um, to help drive the machine learning classifier. So there's that. So definitely it's not using all of them. It's only using the ones that are actually helping it learn, and then helping it learn based on the data that you've provided as the objects you've selected to train the model. Okay. Now, if I take a step back and actually go back to the histograms, we have another question from Marco, as well as a question from Blaze that is talking about this. And so first, the question is, okay, well, if you were just doing a classification with a 1D histogram, and let's just sort of ignore those stripes there and play around just with these objects and their changing colors. And as I said, you can grab this this threshold and it reports to you what that value is, and you can zoom in on the histogram, but why can't you just type it in? And that's one of those things that comes down to implementation, and that we chose this way. We are hearing from you uh, in this webinar setting, as well as in other feedback that we've gotten, that you would appreciate being able to just type in a value. And so that's something that we hope to be able to add in a future release. And so it's not that there is a specific constraint. We would love to be able to add it. It was just one of those things that didn't happen in this first iteration. Okay, so let's see there. And then Blaze had a question about filter 2D. And so again, in this situation, it feels like we have some constraints here, and that is right. Within the 2D histogram, any of the classes that you make, they will have to be these rectangular shapes. And so you can go in, and hopefully you're seeing, as I move it around, the box around, and move my cursor around within the box, you're seeing that it's turning on different modes. And so I can move the whole box, or I can go up and to the edge and change the shape of the box, move the corner, that sort of thing. But again, yes, you are restricted to having a rectangle. And it would be super cool. We definitely agree. I'm sure every single supporter on our team would love to, the ability to be able to circle objects within that 2D histogram. So again, that's something that we're looking into, and thank you for your feedback, because by telling us here, it does help us know what you all are looking for. Let me make sure that I'm answering the other questions, so I have it on another screen that I'm reading. Let's see, what else do we have? Yes, and so Orly was asking a similar question about being able to, are you restricted to squares and rectangles? That's right, and you aren't able to enter in your values. But again, that's something, given that that's a hot topic in the webinar from you all and the questions you're asking. That's good for us to hear. So thank you for that. Let's see. Okay, so Livia is asking, how do I know how many objects I need to select? Okay, so I do think that there is an element of having to play with it. And I know that that's an incredibly frustrating answer to hear, but what I have found personally is that if it's a hard classification for Imaris to learn, or for the um, uh, for the machine learning model to learn that and to be trained on it, then you're going to have to provide even more training data. So if it's something that's taking into account a lot of different aspects, so it's not just intensity, but it's also location, and it's also size and shape, and then you need to make sure that you're sort of covering all of those bases. Sorry, this is very much an American saying, but you're covering all of those aspects within the training data that you're providing to the classifier. However, if you're in a situation like with this data set that we're staring at here, that looks a little funky at the moment, but where, you know, it's intensity. It is mostly intensity, and now it's helpful to to be able to recognize intensity outside of the actual spots, but it is largely intensity. Then you can get away with doing um, less objects for training, and so you will get a feel for that because you do get this automatic feedback once you've trained the model. You are getting an idea of right away of what the results are. And so then you can iterate upon that and adjust it. So if you do find that something is wrong, so either that, let's go ahead and actually go in and do this. So let's clear them, turn these spots back on. So if I go in here and select these and say blue, I got a whole bunch in there, right? And then let's just do a few that are pink and train and predict. And it did a pretty good job, right? I started off with 22 of pink. That's not bad at all. But if I do find that there are some that I'm not happy with, and so maybe I need some more pink ones down here, right? Then all I need to do is go in and select these and then classify them as pink now, and I can go in and train and predict. Now, if I did something incorrectly, you fix it in the exact same way. So if I originally assigned something and made it pink and it doesn't need to be pink, then I can just go in and deselect it. And I could say, oh, I need to make sure that all of this stuff is actually blue. Then I could add that in as a blue object for training and go from there. So I hope that answers your question, Livia. And I think we answered most everybody else's question. If I didn't, Anna, please speak up. I do want to thank everyone for hanging out, and I want to thank everyone for coming to a lot of the webinars. We've been seeing a lot of the same names over and over again. So thank you so much for paying attention and giving us great feedback along the way. And I hope you all can also make it to Sasha's webinar next week. And then again, thank you to the Imaris team for giving all of these webinars. Myself and Anna each gave, I think, 2 of them, and then the rest of the team each gave 1 or 2 as well. And so thank you so much for doing that. And we've been really happy with doing this for you all. So we'll be doing some other interesting things in the next few months. So stay tuned. And yeah, thanks again, everybody, and enjoy the rest of your day and the rest of your week. Take care. Bye-bye.